All right, so I've broken this down into 3 separate topics. The first topic will be running infectious disease-based queries. And then we'll do autoimmune and allergy, and then I'll talk briefly about other types of topics that you might be interested in looking into. In the interest of not, obviously I can't display this at the same time I'm running it, so we'll do one at a time and go back and forth. Sorry if that seems a bit awkward. So the first one I'll look at is what antibody data is from what antibody-specific falciparum epitopes have been defined for humans. All right. Okay, so this is going to be, we can do this from the home page. So we'll type in Plasmodium falciparum. And if I had typed out a little bit more of the foul, the more letters I would have gotten the high node. We were referring earlier to the high node that is the first thing that usually shows up in the drop-down list from the data tree that allows you to get at the majority of the data at once. All right, so I'm going to select that. Let me know if I'm going too fast. All right, and then it's B-cell data, so I'm going to unselect T-cell assays and ligand assays. And I believe the other criteria was in humans. So we'll come down here and use the radio button to select human, and we'll hit search. So along the top, you can see the total number of epitopes for that particular query is 595. Again, you can sort here. Just going to show you some of the functionalities. While that's going, I can show you also that this is a list of antigens, and again, you can sort here by alphabetical order. That'll help you find the one you're interested in very quickly. You can also look at the distribution. In this case, we picked all B-cell assays, but if you had not specified assay, you could look there for that. And again, this is a summary of that data with the records in rows. The reference data shown here. Another cool thing that can be done here is if I sort by assays, you'll get the epitopes with the greatest number of the epitopes from that with the greatest number of assays. In this case, we've got Amerizoid surface protein 3, which is one of the antigens you often see. You can then filter on that particular set of data here and look and drill down. And then also I'd like to show you along the top here. We've mentioned before this shows your filter criteria up here. So we can show you live how you can go ahead and click that off and redo the search. And now you're doing the same B-cell search or antibody responses to Plasmodium falciparum, but you've now excluded the filter for humans, and now it's all hosts at this point. So that shows you how you can easily go back and forth. Are there any questions here for that? Okay. So here's another example. What protective T-cell epitopes have been defined against TB in mice? And then from what antigens? So because this is actually a good time for me to bring up. When you're thinking about your query, so obviously your query is thought of in terms of a sentence, right? what T-cell epitopes have been defined in that. Think about it now that you know a lot more about how we organize the data. You can think about it in terms of the different elements of the things that you're looking for. In this case, we know we're looking for TB, we're looking for mice, we're looking for T-cell, and because you're looking for protection, epitopes that are protective, that's defined in an assay, correct? So that's a challenge assay. So now that you know that you have a challenge assay that you're targeting and you need a finder in order to get to a specific assay, you'll click in here to get to the expanded version of the search panes here. And up here we can start with typing in Mycobacterium. Tuberculosis, and select that. And you'll scroll down to T-cell responses. And here I'm going to type in just generically protection and see what the autocomplete provides for me. So you can choose protection challenge assays. This should be the high node for all of the protection assays. So if you click that, that'll add it to your search criteria. And then we know this was to be in mice. So you can select rodents, but that would also give you a rat. So I would suggest you type in mice. Select that. And then go ahead and hit search. And again, you can glance up at the top to review your input criteria. So here we see 11 epitopes that have been defined in mice. 9 different antigens for Mycobacterium. And let's see, this gives you an idea of how these assays look. So in a mouse host, they administered the epitope, and it was shown to be protective against the organism. So this is the assay antigen here in a challenge assay. And oftentimes in the challenge assay, you will get response frequency. So you can drill down and look at that, and I'll show you. If there's restriction defined, we will capture that here, of course. So let's check out this particular context. We'll drill down into that. This shows you the paper from which the data came from, the epitope information, the antigen, so antigen 85B. So in this case,
how did you get there?
Oh, thank you. So I looking at the summary of the assays here. I scrolled down to just a particular one to show you. So I clicked here on the blue assay ID number. So I clicked there, and that took me to the details of that assay ID. So here's a good example to show you kind of how when we capture this data. So this was a challenge assay. So in this case, black 6 mice were administered this in vivo. So it was administered, the epitope was administered in vivo for the purpose of reducing disease. So that they were doing this as a challenge assay. So the immunogen was the epitope. There's some annotation there, how they were infected. Sometimes they provide dose, that sort of thing. That was a positive challenge assay. There was a restriction defined in that assay, and it was to Mycobacterium tuberculosis. And then that was from Figure 2A, if you want to check that out in the paper. So you can go back to the paper and look that up. All right. Okay. Everybody, am I going slow enough for everybody to follow along? Okay, just raise your hand if I'm going too fast, and I'll slow it. Okay. So, the next sample question. What monoclonal antibodies have been described for dengue virus? And you saw a little bit of how to do monoclonal antibody searches from NEMA. Now we'll do a live one. So because it's a particular type of response, a particular type of antibody, you're specifying the type of antibody in this case. So you'll want to use the expanded, the search panes with the expanded capabilities, which are in. So I'll click search to go in to the results page. And I'll start up here with the organism. Type in dengue. Select dengue, and then I'll come down. whoops, sorry, let me back up. I do this every single time. I'm going to hit reset. This is a specialized search. So I'm going to come up here to the top banner and open up the menu for specialized searches. And I'm going to come on down and grab B-cell assay details. And you'll see when I do that, the system will change the look of these search panes because now it's incorporating all of the details in a collapsed fashion that you can look up. So now here, from here, I'll type in dengue. All right. And then we're looking at monoclonal antibody responses. So we're going to come down to the assay, and we're going to look at assay antibody. So that's assayed antibody. Again, this is jargon, but it's intuitive because it's very explicit. Then you will come down to the purification status of the antibody. And in this case, you can select monoclonal, but you can also look up display libraries because very often they are defining monoclonal reactivity. So you can just click monoclonal, or you can click all three of these, and then you can narrow your search later. For now, I'll click all three. And then I'm going to hit search. Okay, so there's been numerous epitopes defined for monoclonal antibodies against dengue. And of course, there are 4 serotypes for dengue, so this is incorporating all 4 serotypes because I did pick generic dengue virus, so that's reflected here in the number of epitopes you're seeing. So again, because it's a virus, you've got a genomic polyprotein representing the antigen, the protein from which the epitope was derived. And the summary of the assays here. And again, you see this summary. I'll try this. I'll show you this first row. Here you can see that there's negative data and positive data, which you get when you do a specialized search. You get both. This relationship here, showing that it was a taxonomic sibling, demonstrates cross-reactivity. So people who work with dengue know the importance of cross-reactivity and also issues related to cross-reactivity that are not so great. So that's an important thing that you can look for. And then again, if we were to look at one of those particular assays, say I'm just scrolling down to one of the assay IDs and I'm going to click on it, in this case 504, sorry, 10504. And then I can drill down and look at the details of the epitope itself, the strain that it came from, serotype and strain, the host, how the host acquired its immunity, in this case being administered the virus itself, and then reactivity, very strong reactivity in ELISA. In this case, it was actually, they measured KD, and there's a quantitative value for that. We do capture quantitative values for antibody responses of affinity. When that is performed, we can capture that to reflect overall affinity of the antibody. We also capture X-ray crystallography. We have a person who's dedicated to capturing that data too, which is wonderful.
Can I ask a question?
Sure, yeah.
Why is the immunome browser disabled for this protein? Why does it grayed out?
Let's look.
Go back to the previous window. I'm under antigens. Why does that link not work for this protein? Is that something related to the source of the data, or?
Pardon?
We don't have the immunome browser functioning for the specialized searches at this point, and that's something that we're working to fix.
Yeah,
soon.
Yeah, it's a complex. There's complexity to the answer, but we'll just leave it as it's not engaged yet, not functional from this particular type of search. But there's nothing stopping you from looking at, and I can show you this right now. This is a good. You can perform the same search looking at antibody responses, and let me do that since we have time. Let's see. it's a way to work around right now while it's not working. You can run that search. And well, I was going to download the spreadsheet so I could show them where in the spreadsheet they can find monoclonal antibody designations and look at it that way. And then to find the antigens from which, yeah. But we can do that too here. So here, if you were to perform the search from here, not without the filter of specifically monoclonals, you can do it from here. And then what you can do is, in fact, let's do that. And while that's running, so if you were to for those of you who haven't downloaded one of the spreadsheets that contains the data from your query, this is the way you would do it. I often do this when I highlight the row that is the column head so I can see, and then I come here and hit filter so that I can filter on those. And this is, you can do this if you haven't done it before and familiarize yourself with the organization, but it's the same thing. The first part is about the first several columns, identify the reference, and then moving this way, the epitope, and then the assay, and then the antigens in that direction. So if we come over here in this B-cell spreadsheet, so you'll see here, there's a column that indicates whether it's monoclonal or polyclonal, so you can filter on that. There. And also other details we can capture if they provide, which is the name of the monoclonal antibody and the isotype of the monoclonal antibody. And then, let's see, and how many do we have there? 1,700. That's a lot of data. Then if you were to go back this way, because you've filtered on that, you now have the antigens from which those were, for which those are defined here in this column. This is the epitope antigen name, the epitope source antigen name here. And then what you can do, okay, so here we go. So this, now that that came up, for this, not to jumble around too much, sorry, I apologize. So just the strictly dengue virus and B-cell responses is what's mapped to this here. And then in the interim, what you can do is you can use the information from the spreadsheet here of the monoclonals and use this data table to match to them if you find ones of interest. So that would be a quick workaround. But also, there's information to be gained from the just doing B-cell data, a general immunome plot. But it's good that you pointed it out. Thank you. All right. And then the last infectious disease-specific one is what tetramers have been defined in the context of influenza virus, and then what alleles. So again, we know that we have a specific assay in mind, so we'll want to go into the search panes with the expanded functionality of the finder. And here we'll first type in influenza, since we'll go top down. Influenza A virus. Then we'll unselect B and ligand and type in Tettemer. All right, so the results show nearly 400 epitopes. And the antigens. So you can sort on these to get alphabetical order. And this is from most of the proteins, if not all, 8. All right, and then to look at the alleles, you would come to the assays page because it's captured in the context of the assay. And you can, again, sort on this column, which is MHC restriction, and look at that. And then you can also download this in Excel as a spreadsheet and sort on that column, the same column that would be in the spreadsheet, and get at all DB, all A2. Alternatively, you can refine this search here and look for particular. And then I'll redo the search on just DB. And that should narrow it down quite a bit. Okay, so yeah, I think it cut the number of assays down by quite a bit, maybe by a third or so. I can't remember what the total number was. I'll also make a suggestion if you are interested in tetramers as assay antigens to be used, and you're coming here, it might be worthwhile looking at the higher node, the serotype, because that way you can see what's been done and what peptides. For example, you might find that you might have in the literature something that's been tested at the level of A1, but not a specific A1 allele. So you'd still be interested in that, but if you typed in your specific allele, that wouldn't come up necessarily. So I would say start at the higher node, specifically for Tettemer. We have a lot of Tettemer data in here now. It's been an ongoing project to capture Tettemer data as of recently.